All right.
All right, good morning, everybody. It's a real privilege to be here. I appreciate the invitation. And I just want to call out to the family group that these family stories are incredibly moving. So I just want to congratulate you on helping to bring the human situation to the researchers in such a powerful way. So I'm going to give a brief talk today, and I want to sort of touch on a couple of these points. I want to talk a little bit about what the mission of the Simons Foundation and SFARI is. I want to talk about the goals for Simons Searchlight, both for families and researchers. Just extremely briefly mention what we're learning from the registry that we've been putting together, and then have a quick look to the future for some Simons projects. I see this works like every third time. Okay, so a quick background on the Simons Foundation. It's a private foundation based in New York City, founded by Jim and Marilyn Simons. It has a very large endowment of about $3 billion and an annual spend of somewhere in the $400 million range. The part that's most of interest to this audience is called the Autism Research Initiative, or SFARI, Simons Foundation Autism Research Initiative. I'm the deputy director of that program. And our mission is pretty easy to say, pretty difficult to carry out, but their mission is to improve the understanding, diagnosis, and treatment of autism spectrum disorders by funding innovative research of the highest quality and relevance. Our budget in the last few years has been in the range of about $75 million a year. And since we launched in around 2003, we've funded about $380 million of external research support, much more if you include the research that's been sort of coordinated directly through the foundation. We've supported over about 450 investigators. For anyone who wants more information about SFARI, I would send you to safari.org, which is our science team webpage. We also, if you're into that kind of thing, have a pretty active Twitter feed. And then if you're can't remember to check the website, we have a newsletter that comes out about once a month that has all sorts of information about funding opportunities. Okay, let's get into it now. Sorry. So what specifically about SCN2A? Well, first, we're a direct funder of basic and clinical research. A quick calculation over the last couple of weeks is that we've spent somewhere in the range of $3 million over the last couple of years directly funding work having to do with SCN2A. One of our primary goals, though, is to provide resources for research. So that includes animal models, iPS cells, and I'm going to talk a little bit later about iPS cells, with the goal of lowering the barrier of entry for other researchers to come into the field. So one of the early goals of SFARI for autism and neurodevelopmental disorders writ large was try to attract new researchers by not only providing funding, but also providing resources to make it much easier for people to come into the field. I think one of our important roles has been a convener. So we've held a lot of workshops. We have all sorts of investigator meetings. We had a workshop a couple years ago that I'm especially proud of that resulted in this paper that Stefan Sanders and Kevin Bender and others took primary lead. Many of the researchers in the room were at this workshop, including Leah. And so this sort of was a workshop directly focused on SCN2A. It hasn't been mentioned today yet, but I just want to also call out that largely due to the efforts of Leah, but we've been helping to support this. We have roughly quarterly calls that are open to any investigator, and I assume families around the world. These calls are investigators report on updates and stuff. And I would just want to shout out to everyone, if you're not aware of these, get in touch with me or Leah and we'll put you on the mailing list, but these have been a very productive ways. I think one of the most important things in a really complicated field like this is that we're a neutral player. The Simons Foundation has no interest in taking intellectual property. We're a convener and a supporter, but we consider ourselves a fully neutral player. There was a talk yesterday to the families. So for researchers who are not aware of this, we're also supporting a program called Autism BrainNet, where we're helping to collect and distribute brain tissue from postmortem brain tissue. We hope this will be also very useful to the research community. And now I'm going to jump and just spend a few minutes talking about Simons Searchlight. So Simons Searchlight is a program within SFARI, and the goal is to collect detailed medical and guess can I walk around here? Detailed medical and behavioral histories on, along with biospecimens from individuals around the world to synthesize this information and provide results back to families, to freely share the data and sample with qualified researchers, and qualified really just means that you're a researcher acting in good faith. And then to connect participants around the world and provide essentially a better understanding of specific disorders. So the project has changed substantially over time. Some people in this room are involved really right from the beginning, but it started in 2011 and it focused on families that had one specific genetic change, which was a change 16p11.2. It's a copy number variation. The first participants traveled to study centers and provided medical, behavioral, and neuropsychiatric data to researchers. Today, we've expanded this program very substantially to involve over 100 different genetic changes that can increase the risk of autism and related neurodevelopmental disorders. I'm showing you sort of the top, I don't know what this is, 8 or something like that. You'll see that next to these copy number variants, which are very prevalent, SCN2A is among one of the largest groups of genes. There's 100 of these. They fall off pretty significantly, but we're really proud of the fact that we've helped to sort of bring together so many families along with the Family SCN2A Foundation. So what do you do as a participant? You provide medical behavioral histories and samples. There's a roughly annual update. Samples are collected locally or at family meetings. So I want to do a shout out. I know many of the parents are in a different session right now, but there's going to be a blood collection session over the next couple of days here. So families who are here and have not signed up, I want to call out to the people at the table right when you leave the door here, and they'll be happy to help sign you up. You become part of this network. There's a lot of social media components to help connect families and provide durable and reliable information. This is just a snapshot of the type of information that's collected. In everybody's packets at registration was a book that has much more information about this, so I'm going to breeze through this quite quickly. This is really critical, and again, this is mainly directed to families, that the project has evolved from an older project that we called Simon's VIP to Searchlight, and it is pretty important for participants to go in and update your information. It's essentially a one-click thing, but it is important that you do that. So a little bit of information about the registry. This gives you some information about where families are in this process. There are 100, I can't read it from this thing. There's something in the range of about 180 families. This is also in the booklet that was handed out. And you'll see that there are families at different stages of this. There's families who are registered, consented, those with lab reports. It's extremely important that we have a team at Geisinger and both at Simons that are sort of going carefully through the lab reports to make sure that families are signing up for this program really have what they think they have. There's sort of a medical history phone call that takes out, and then when all of this data is deemed sort of clean and reliable, it's released to researchers around the world. If you haven't looked at this database as a researcher, I would encourage you to do so. This obviously you can't read here, but to give you a sense of the range of different types of mutations in families that are now in the database. They include missense mutations, likely gene disrupting mutations, splice site mutations, deletions, etc. Sort of a very quick peek at the medical history data. There are many more pages of this in the booklet that was handed out, but the important caveat is at this point, this data is not divided by the particular mutations. And I think that's obviously going to be a really critical thing because some of these phenotypes seem to be specific to the particular mutations. So, but this is sort of a bird's eye view of some of the responses to the medical history intake. There's some information here. This is going to be, I think, hard to read in this kind of format, but about different types of seizure medicines. Again, I think this data will start to become particularly useful when we have even larger ends and we can sort of segregate this importantly to the different type of mutations. For researchers who are interested in looking at this data, and I would encourage everyone to try to do so, all of the Simons resources are distributed through a centralized website called SFARI Base. It's relatively trivial to sign up. You have to sign some very simple things saying you're not going to try to identify individuals and that kind of stuff, and then you can get access to this. Okay, so I want to just take a couple minutes and sort of look to the future. Always something dangerous to do, but I think it's sort of what we have to do thinking forward. So I'm going to talk very briefly about a new project at Simons called Spark and how that relates to what we're doing in the room here today. I'm going to talk a little bit about our efforts in induced pluripotent stem cells, more commonly known as iPS cells, and then very briefly about some new methods for phenotyping that we've been keeping our eye on at Simons and helping to develop. So the first project I just want to make sure everybody knows about is called Spark. This is a sort of the evolution. Simons has become well known for assembling different type of cohorts. The first one was the Simons Simplex collection, which had an enormous effect on understanding the landscape of autism genetics. The next one was Simons Searchlight, which I think we know about here. And this latest cohort is called Spark. So the idea, this was launched a couple of years ago, is to recruit and engage a community of about 50,000 individuals with autism and related neurodevelopmental disorders and their families. And one of the main goals is to ensure that this is a recontactable cohort so that researchers around the world can access these patients and recruit them for various studies. It's very different from other projects that we've done in the past. So to register, people create an account online, they invite family members, they spit into a small tube that we send, and basically that tube goes off and gets exome sequenced. So this is going to be an enormous resource of exome sequencing of mother, father, and kid with autism or related neurodevelopmental disorder. And I just want to highlight, this is a pretty extraordinary number. This is a recruitment as of July, so just last month. So there are 27,000 individuals that are consented, and critically almost 18,000 completed trios. So that's mom, dad, and proband. So the only point I want to make with this very simple slide is when this exome sequencing is complete, which is going to be relatively soon, it's very likely that there are going to be a large number of new families with SCN2A and other mutations that are going to be presumably joining Searchlight and the SCN2A family group. So I think this is going to be a really interesting resource moving forward. I want to say a quick word about induced pluripotent stem cells. You heard a little bit about this from that fascinating gene therapy talk that we just heard. So access to the cells that we think are relating most directly to epilepsy and autism, which are brain cells and all the associated cells, it's very hard to get access to those, not surprisingly. There's this extraordinary technology that's relatively new where you can take somatic cells, so skin cells, or in the case that we're pursuing most aggressively, blood cells, and you can actually transform them back into what are known as pluripotent stem cells, which then have the ability to be coaxed into forming neurons or glia or other kinds of cells in the brain. This is an extraordinary, opens up some extraordinary possibilities of studying human neurons that carry all of the richness of the human genetics, and I think is going to be extraordinarily important for disease modeling and drug screening. There's also been some extraordinary advances in actually growing what are some people refer to as organoids or mini brains in the dish, where you can actually create sort of early nervous systems to study in a slightly more systems context. But there are an enormous number of challenges here. It's very hard for most researchers to get access to large numbers of very well-defined patient cells with accompanying phenotype data. The access to appropriate controls has been a really big issue as well. People debate whether familial controls or so-called isogenic controls are most critical. And access to high-quality iPS cells has been a challenge due to the technical variability. So it's possible to do this work in your own lab, but it tends to be very difficult to do at a very high level of technical ability, and there's really some extensive quality control steps that are critical, and of course funding for all of this. So pretty much, so SFARI over the last year or so has been gearing up for a pretty major project where we're going to be taking many of the blood samples that are in the freezers, thanks to many of the families, like the families here at SCN2A, and through an organization that we're going to contract with, basically making large numbers of iPS cells and then distributing those at no cost, essentially, to researchers around the world. So I want to sort of encourage people to keep their eyes out for this. SCN2A is going to be among the first batch of cells, largely because of the demand and because we have so many of these. So I think this is going to be a really important research resource for the community. And then just in the last 2 or 3 slides, I want to sort of bring up an issue that I've been thinking about for many years, and I think Simon just could also try to make an impact here. And this has to do with the new methods of phenotyping. So we have all these families now that have been identified or in these databases, and still relatively sparse phenotype information. So phenotype, for the most part, has been subjective and episodic. So when people go into the doctor's office or into a research clinic, provider-based, reactive, and distant from the core biology. So many of the instruments that we use, which are sort of the best things out there, are still quite distant from the core biology that we as neuroscientists think of. And this has often led to people making decisions based on sort of population data. And I think where we'd like to be in the future is thinking about more objective and continuous measures. Those that allow us to look longitudinally. Especially ideal would be measures that are passive, ubiquitous, unobtrusive, so they can be used in the real world, and proactive, that are closer to the core biology, allow us to identify problems early and take advantage of maximum brain plasticity, allow us to think more in terms of a diagnostic continuum as opposed to binary diagnoses of autism or epilepsy or not, for example, and allow us in particular, and this is, I think, going to be really important for clinical trials, to measure in a personalized way sort of deviation from personal baseline as opposed from some group averaging. I think this is going to be critical. I'm just going to show one slide. Oh, and so why now? Like, why are we thinking of this now? I think there's been extraordinary advances in digital electronics and chemical engineering and signal processing, most directly deep learning. As many people in the room know, there have been major investments in some of the largest Silicon Valley companies in this area. I've been to several meetings over the last few years where I think something is going to happen. It's sort of in progress. And I just want to give one example of sleep. It's obviously a very common issue, a problem in autism and epilepsy and related disorders. And the current gold standard for measuring sleep beyond sort of a simple questionnaire asking if someone's sleeping well or not is overnight polysomnography. I've actually done this. If anyone's done this, it's an incredibly artificial thing to do. You go into a sleep lab, you're wired up in all sorts of crazy ways, and then you're supposed to have a relaxing night's sleep. It doesn't also capture circadian rhythms, which are critical to understanding sleep, and it tends to be sort of an n=1 or n=2 thing where you go into a sleep lab for a day or two. This is not a product endorsement for either Fitbit or for the Dream headband, but it's just sort of saying that there are a number of technologies that are coming online that allow us to measure sleep. And I'm using just as one example. I think there are many other examples here, but sleep and even EEG in a much more natural setting where we might be able to, at low cost, to get longitudinal multiple data. So anyway, I just want to sort of stop with this slide here. I just wanted to point out that Simons has been very interested in this and trying to roll this out. I have a sense that if we sort of move forward in a big way with this, that we might do this in the identified cohorts like the SCN2 group and some of the other groups that we're working with to try to, I think these are the perfect sort of first test beds for this kind of data. So I'm going to stop there, and then I guess we'll take questions after the next talk. So thank you.